Neuroscientists study the function of the brain by investigating how neurons in the brain communicate. Here, changes in the electrical activity of a single neuron are evoked upon application of an experimentally controlled conductance using the dynamic clamp technique. First, thin rat brain slices are cut and stored in an incubation chamber filled with artificial cerebrospinal fluid. A slice is then transferred to a patch clamp recording rig. A neuron is selected and then patched with a recording electrode. Using a dynamic clamp computer connected to the recording amplifier, an NMDA receptor conductance is applied into the cell. The resulting recordings show a burst of action potentials, suggesting that activation of NMDA receptors is one cellular mechanism through which dopaminergic neurons can generate bursts.
The main advantage of this technique over current methods like direct current injection or pharmacological treatment is that we're able to precisely determine the effects of activation of an experimentally defined receptor on the electrical activity of a living neuron.
After quickly extracting the brain from an anesthetized Bragg-Dolly rat, cut 240 micrometer horizontal brain slices using a vibrating microtome. As the slices are cut, use a transfer pipette to place them in a 32°C incubation chamber filled with artificial cerebrospinal fluid until it is time to perform recording experiments. The intracellular recording rig consists of a microscope with objectives set up for gradient contrast imaging, a monitor, a perfusion system, a multi-clamp 700B amplifier, an analog-to-digital converter,